Welcome to Bored to Sleep, where either you'll get some rest or learn something new. Today, we'll be falling asleep while learning about milk microbiota and milk in the modern world. Settle in, close your eyes, and prepare to rest. Let's begin.
Milk microbiota, characterization methods and role in cheese production. Highlights. Milk microbiota has a pivotal role in the cheese properties, flavor and aroma. Cheese-making technologies impact on the dairy microbiota composition and activity. Ripening selects microorganisms responsible for the cheese flavor and aroma. Omics technologies enable a comprehensive investigation of the cheese microbiota. Abstract. Milk is a complex body fluid aimed at addressing the nutritional and defensive needs of the mammal's newborns. Harbored microbiota plays a pivotal role throughout the cheese-making process and contributes to the development of flavor and texture typical of different types of cheeses. Understanding the dairy microbiota dynamics is of paramount importance for controlling the qualitative, sensorial and biosafety features of the dairy products. Although many studies investigated the contribution of single or few microorganisms, still there is some information lacking about microbial communities. The widespread of the omics platforms and bioinformatic tools enable the investigation of the cheese-associated microbial community in both phylogenetical and functional terms, highlighting the effects of the diverse cheese-making variables. In this review, the most relevant literature is revised to provide an introduction of the milk and cheese-associated microbiota, along with their structural and functional dynamics in relation to the diverse cheese-making technologies and influencing variables. Also, we focus our attention on the latest omics technologies adopted in dairy microbiota investigation. Discussion on the key steps and major drawbacks of each omics discipline is provided along with a collection of results from the latest research studies performed to unravel the fascinating world of the dairy-associated microbiota. Significance. Understanding the milk and cheese-associated microbial community is nowadays considered a key factor in the dairy industry, since it allows a comprehensive knowledge on how all phases of the cheese-making process impact the harbored microflora, thus, predict the consequences in the finished products in terms of texture, organoleptic characteristics, palatability and biosafety. This review collect the pioneering and milestones works so far performed in the field of dairy microbiota, and provide the basic guidance to whom approaching the cheese microbiota investigation by means of the latest omics technologies. Also, the review emphasizes the benefits and drawbacks of the omics disciplines, and underline how the integration of diverse omics sciences enhance a comprehensive depiction of the cheese microbiota. In turn, a better consciousness of the dairy microbiota might result in the application of improved starter cultures, cheese-making practices and technologies, supporting a bio-safe and standardized production of cheese, with a strong economic benefit for both large-scale industries and local traditional dairy farms. Introduction. Cheese is one of the most consumed milk-derived products worldwide. Its acceptability to the final consumer is largely dependent on the specific organoleptic properties, including flavor and aroma. Altogether, these properties are strongly dependent on the myriads of compounds and molecules constituting this product, such as fatty acids, volatile organic compounds, VOCs, amines, ketones, free amino acids, phenols, alcohols, aldehydes, lactones, sulfur compounds etc. 1,2. In turn, these molecules are linked to several factors, including type and altitude of the pastures, technological aspects of the cheese-making process and ripening conditions. 3, 4, 5. Besides, a pivotal role in determining the specific cheese peculiarities is played by the milk-associated microbiota. Indeed, the complex composition of milk, and all its derivatives during the subsequent processing steps, provides a rich nutrient source, enabling the optimal growth of a wide array of microorganisms of diverse origin. Here, the microbial metabolism of a selected number of colonizing bacteria, that is lactic acid producing bacteria, lab, transforms the milk components, mainly carbohydrates and proteins, into secondary products that could further be used as a substrate for the growth and the metabolism of other microorganisms. These, in turn, provide a strong contribution to the composition of the final product. 1,5,6. The whole cheese-making process is a finely tuned series of microbiological and biochemical events where microorganisms play an active role in determining both cheese composition and the structure of the hosted microbiota. On the other hand, during the cheese-making process, the cheese microbiota can be influenced by the physicochemical stress, pressure originating from the different technological phases. 1,7. The aim of the current review is to provide a comprehensive overview on the dynamics of the milk microbiota in relation to the diverse cheese-making processes and technologies, with a specific focus on both big and small ruminants. The review discusses the methodological aspects and drawbacks of all mixed disciplines, on the attempt to provide guidance to the growing number of readers approaching to the fascinating world of milk and cheese microbiota. A discussion on the cheese microbiota members and its metabolic contribution for the development of characteristics, flavors and aromas is also provided. 2. Milk microbiota, source and factors shaping the microbiota composition. Milk-associated microbiota accounts for a high diversity of microorganisms, with bacteria being the most represented, followed by molds and yeasts. 8. Both gram-negative, for example Pseudomonas, Flavobacterium, Acintobacter, Aeromonas, and various Enterobacteriaceae, and gram-positive, for example Lactobacillus, Lactococcus, Bacillus, Clostridium, Staphylococcus, Micrococcus, Streptococcus, Microbacterium, Corynebacterium. Families are commonly identified in the microbiota composition. 1,4. Milk microbiota composition is rather dynamic and linked to several host-related and environmental-related factors. In the following sections we will review the microbiota composition of milk from the major mammalian species, as well as how diverse environmental factors could shape the milk microbiota composition and the cheese-making process. 2.1. The cow milk microbiota. In 2017 European Union produced 170.1 billion tons of raw milk, of which 97%, that is 165 billion tons, was cow's milk, whereas the remaining 3% is shared among mammals other than cows. Eurostat, 2017. Several studies have been so far performed to assess the cow milk microbiota composition. Although the adoption of diverse investigation methods and the influence of diverse factors, both endogenous and environmental, hinder a direct comparison and integration of the obtained results. The typical composition of the cow milk microbiota registered a heterogeneous composition featured by the high abundance of lab such as Lactococcus spp. 101 to 104 CFU per milliliter, Streptococcus spp. 101 to 104 CFU per milliliter, Lactobacillus spp. 102 to 104 CFU per milliliter, Leuconostoc, 101 to 103 CFU per milliliter, and Enterococcus spp. 101 to 103 CFU per milliliter, 9,10. Besides, bacteria with a variable load such as the psychotropic Pseudomonas, Acintobacter and Aeromonas have been also described in the cow milk microbiota. 9,11. Moreover, recent investigation based on more sensitive methods compared to the traditional ONISH revealed the presence of anaerobic bacteria such as Bacteroides, Faecalibacterium, Prevotella, and Catanobacterium, whose origin is most likely linked to fecal contamination events. 9. 2.2. The goat milk microbiota. Goat milk production in the EU concern over 1.5% of the total milk production. Eurostat 2017, with a steadily increasing trend, most likely due to its widespread application as an alternative to cow's milk because of the higher digestibility and lower allergenic potential. Goat's milk is typically used for cheese production, especially in small dairy industries of the Mediterranean regions and southeastern European countries. From a physicochemical perspective, goat's milk is featured by an elevated level of iron availability, small fat globules and a high content of fatty acids. Altogether, these properties represent a characteristic milieu selecting for a dynamic microbial community typically composed by labs such as Lactococcus, 106 CFU per milliliter, Lactobacillus, 105 CFU per milliliter, Leuconostoc, 103 CFU per milliliter, and Enterococcus, 102 CFU per milliliter, 9,12. Following these bacterial genera, Enterobacteriaceae and Micrococcaceae are commonly isolated. 12. In the study from Callan et al., Corine bacterium, Brachybacteria, Polyphilic Gogalococcus cicrophilus, Salinicoccus spp, Dietzium aries, Exigua bacterium, Ornithinococcus spp, and Haelocheguensis were also identified by culture-dependent methodologies. 9,13. 2.3. The sheep milk microbiota. Sheep milk is rarely consumed in a direct manner, however, its production accounts for approximately 1.3% of the total milk production. Eurostat, 2017, 9. Nutritional composition of sheep milk is influenced by the geographical location and, as a consequence, the composition of the harbored microbiota varies. Lactic acid bacteria are highly present. Mesophilic bacteria range from 102 to 106 CFU per milliliter. Psychrotrophic bacteria range between 102 and 104 CFU per milliliter. 9. Studies on sheep milk microbiology, 14,15, link the higher fat content to the greater load of lab, coliforms and molds. Streptococci and Staphylococcus aureus count were respectively increased and decreased in milk with reduced nutritional value and a higher acidity. 9,15. Other bacteria rarely found in sheep milk are the health-affecting Salmonella, Escherichia coli and Clostridium perfringens. 14. 2.4. The buffalo milk microbiota. Buffalo milk, as a fresh product, has little market in the European countries in comparison to its derived buffalo mozzarella cheese. Recent investigation on the raw buffalo milk showed Lactococcus spp. and Acinetobacter spp. as the dominant bacteria, accounting for 30% and 21% of the total bacteria composition, respectively. Other abundant bacteria species include Pseudomonas spp. 20%, Streptococcus macedonicus, 10%, and Lactococcus lactis, 10%. A number of other low-abundant microorganisms include Broccothrix spp, Carnobacterium spp, Chrysiobacterium spp, Clostridium spp, Corine bacterium spp, Enterobacteriaceae, Gammorproteobacteria and Halonella spp. 9. 2.5. Environmental factors shaping the milk microbiota composition. In addition to the milk-producing species and other intrinsic animal factors such as physiological status, number of milking sessions, and endogenous antimicrobial enzymatic system, 5,6, the environmental factors are undoubtedly among the most important sources of variability for the milk microbiota composition. Fresh milk includes a wide variety of microorganisms of animal origin. The udder surface represent the first source of milk contamination with bacteria belonging to the Phylloactinobacteria, Firmicutes and Proteobacteria, 5,6. Common contamination source of these bacteria is also represented by the milking machine, bottles, storage tanks and all other milking equipment and accessories. 11. With the purpose of evaluating the milking hygiene, several studies have demonstrated how cleaning, disinfection of the milking paddock and milking machines can impact both raw milk microbial load and microbiota composition. 5, 6, 7, 8. Other relevant factors affecting the raw milk microbiota composition include the type of forage, grass, silage and hay, fed to animals, the hygienic conditions of animals, and milk storage. Previous studies that aimed to provide the microbial profile of the diverse pastures reported that a high amount of Enterobacteriaceae, corine-form bacteria, Staphylococci, and yeasts along with a reduced count of lab have been observed in grasslands. 8. Differently, studies on silage showed the presence of different lab representatives such as Pediococcus pentosessus and various lactobacilli together with the reduced loads of spoilage or pathogenic bacteria. For example, coliform bacteria, Enterococci, yeasts and molds. Hay was found to be a source of Actinomycetaceae and other gram-positive bacteria, also gram-negative bacteria and, among fungal species, Eurotium sp. were found Enterococci. 8. Bonizzi et al. Investigated the raw milk microbial composition in relation to diverse pastures altitude and over 2 consecutive year repetitions. Results highlighted the presence of diverse microbial profiles between samples collected at the diverse altitudes, as well as between the first and second year collection, suggesting a strong effect of both pasture's altitude and weather conditions on the raw milk microbiota composition. 16. In a similar study, Lee et al. Investigated the milk microbiota and its dynamics over time by sampling raw milk at each month of the year. This study describes a strong effect of the weather conditions on the milk microbiota composition, with an increased abundance of Pseudomonas, Propionibacterium and Flavobacterium associated with lower temperatures. Moreover, a negative correlation between total bacterial count and Propionibacterium and Pseudo-Alteromonas was observed, suggesting a potential role of these last two genera in preventing milk spoilage and milk microbiological quality maintenance. 17. Hygienic conditions have also great importance. Indeed, animal feces contamination is among the major source of heterogeneity of microorganisms, including harmful bacteria. For example, Enterobacteriaceae, coliforms, spoilage bacteria. For example, butyric acid producing Clostridia, Psychrotrophs, yeasts. For example, Candida spp, Saccharomyces spp, Lab and Bifidobacterium, 1,9,18. Milk storage and refrigeration effects have also been extensively investigated in relation to the raw milk microbiota composition. Psychrotrophic bacteria such as Pseudomonas spp, Bacillus spp, Acinetobacter spp, and Enterobacteriaceae are common components of the milk microbiota. This bacterial load is highly variable in raw milk, from 10% to 75% of the total bacterial count, depending on the hygienic conditions. 19. Refrigeration of milk affects the composition of the associated bacterial community, since it has a bacteriostatic effect on the only mesophilic and thermophilic bacteria, promoting the selective growth and proliferation of the psychrotrophic ones. In turn, increased abundance of the psychrotrophic bacteria is linked to increased levels of exogenous heat-stable proteases and lipases. Although minor quantities of these enzymes are desirable in cheese industry for the development of a characteristic flavor, excessive amounts lead to milk spoilage and off-flavors development. For example, bitter flavor, rancidity, even after raw milk treatment, for example pasteurization. 1,2, 5, 6, 7. 3. Milk pre-treatments for cheese making and their effect on microbiota. Typically, cheese has been produced by direct curding of the whole raw milk without any preventive treatment. Still today, the great majority of the artisanal cheeses is produced by using raw milk, whereas most of the commercial and large-scale produced cheeses expect a milk treatment to enabling the manipulation of the milk characteristics, the standardization of the milk quality and lowering the milk microbial load. 20. Although the presence of some beneficial and health-promoting bacteria, milk microbial growth is commonly considered as an index of poor microbiological quality. 21. Since the steadily increasing interest in food safety, the European Union Regulation EC 853/2004, aiming to contain and to reduce foodborne outbreaks, set the limit of total plate count, TPC, at 30 degrees Celsius of bacteria in cow's raw milk at 105 CFU per milliliter and a somatic cell count, SCC, at 4 times 105, whereas for the other mammals species, including goats and sheep, the TPC limit is raised to 1.5 times 106 CFU per milliliter. To date, several approaches are used to lower microbial load and guarantee milk security. High pressure and heat treatments are the most commonly adopted practices in both small and large-scale dairy industries. 3.1. Raw milk is a potential threat to human health. Despite being collected from the only apparently healthy animals, pathogenic bacteria may directly originate from asymptomatic animals or through contaminations occurring in the diverse phases of milk collection, storage and processing. 17,22. Pathogens identified in milk samples are strongly dependent by the animal species. A detailed review of Veras et al. Lists the major human pathogenic microorganisms identified in the raw milk of diverse mammals from several countries all over the world. 21. Among the total bacterial diversity, goat milk comprises a variable abundance of Listeria monocytogenes along with verocytotoxin-producing Escherichia coli, VTEC, 19, 23, 24, 25. Studies have also demonstrated the common presence of the zoonotic Mycobacterium of subsp. Paratuberculosis, 26. Staphylococcus aureus contamination is often found to a wide extent, 21,27, and its ability to produce enterotoxin has an important safety concern. Here, staphylococcal enterotoxin A, C and D, C, SCC and ST, respectively, are generally the most relevant from a clinical and food safety point of view, 17,28. Following the Q fever outbreaks in goats and sheep registered in France, 2002, and the Netherlands, 2007, monitoring of the Coxiella brunetii in goats' raw milk revealed the presence of the bacteria in milk from animals that have had miscarriage around a period of 3 months, 29, whereas in a DNA-based investigation performed by Van den Brom et al., about 1/3 of the investigated samples appeared to be positive to Coxiella brunetii contamination, 30. Similarly to goat milk, raw milk from other mammals is also associated with the presence of human pathogenic and/or potential pathogenic ones, 21, posing the need to adopt appropriate strategies, especially in large-scale industries, for an efficient reduction of the milk microbiological hazard. 3.2. The effects of high-pressure treatments on milk microbiota. High-pressure treatment of milk is primarily applied to reduce the original raw milk microbial load. This treatment has a variable efficiency, with gram-positive bacteria being more resistant than gram-negative ones, and endospores being more resistant than vegetative cells, 26,31. Several high-pressure treatments have been investigated over the years. Drake et al. Applied cycle high-pressure treatments with a maximum pressure exposure of 586 mPa, 5 degrees Celsius for 15 min and registered a 4-log reduction of Escherichia coli and 6-log reduction of Staphylococcus aureus, 32, whereas 3-log reduction of Salmonella Seffenberg and Listeria monocytogenes was achieved by applying, at room temperature for 15 min, 310 and 379 Pa respectively, 33. Nevertheless, high-pressure treatments application is restricted due to the inability to apply it to large-scale dairy industries. Also, high-pressure treatment negatively affects the cheese-making attitude of the milk, since reduces sizes of the casein micelles and denatures large proteins such as beta-lactoglobulin, 29,31,34. 3.3. Milk thermization as a strategy to control milk microbial quality. Thermization is likely the oldest and most commonly used strategy to improve the microbiological quality of the milk. It consists of a sub-pasteurization process where milk is usually heated up at 63 to 65 degrees Celsius for 15s and subsequently cooled and stored at refrigerated temperature, 4 to 7 degrees Celsius, to preserve the microbiological quality. The heat application succeeds in the reduction of the psychotrophic bacteria, for example Pseudomonas spp, and some human pathogenic bacteria, but does not ensure the achievement of the public health requirements as pasteurization does. 2. Raw goat's milk thermization at 60 degrees Celsius for 30s showed a significant reduction of Enterobacteriaceae, coagulase-positive staphylococci, and Listeria until safe levels, 35. However, thermization does not prevent the spore germination, for example from Bacillus cereus, so contaminated milk can still represent a health threat. 2. Unlike traditional cheeses, most commonly produced by raw milk, the majority of cheeses produced on a large-scale industry are obtained from pasteurized milk. Pasteurization is usually obtained by means of heating up the milk to 72 to 74 degrees Celsius for 15 to 30s, high temperature short time, HTST, but small dairy producers still use the low temperature long time, LTLT, process with an overall vat milk heating temperature up to 65 degrees Celsius per 30 min. This process is supposed to ensure product safety by destroying pathogens. Nevertheless, in order to inactivate heating-resistant endospore, other treatments, such as physical removal or chemical inhibition, might be required. 4,17,33. Investigation on the microbial dynamics of raw, pasteurized and pressure-treated milk cheeses from goat have described significant differences in the TPC of psychotrophic bacteria, Enterococci, Micrococcaceae, Enterobacteriaceae and Lactobacilli, whereas no differences were observed between thermic and pressure-treated samples. 5. 3.4. The effect of milk treatment on microbial shape. Besides reducing the pathogenic population, milk treatments exert a deep inhibition of the indigenous non-pathogenic microflora. Indigenous milk microflora, along with the traditional tools and manufacturing practices, is considered one of the key elements of the organoleptic characteristics such as flavor, taste, texture. 32,33. Moreover, bacteriocins and other antimicrobial compounds of bacterial origin, along with the several antagonistic biomolecules present in the raw milk, for example lactoferrin, lipoproteins, lysozymes, immunoglobulins, etc., are synergistically active in preventing pathogens' growth. Nevertheless, pasteurization inactivates or reduces the available concentration of these compounds. 35,36. Dumatrascu et al. Reported a significant inactivation of lactoperoxidase in the raw milk of goat, sheep and cow after thermic treatment at 70 to 77 degrees Celsius, indicating an important lack of the naturally occurring milk defense system. 37. Van Gysel et al. Highlighted an increased growth of Escherichia coli and Staphylococcus aureus in human pasteurized milk than unpasteurized ones, most likely due to the inactivation of the native antimicrobial biomolecules. 38. Additionally, raw milk microbiota includes a high diversity of lab and other microorganisms active in preventing human pathogen growth. 1,5,17,22. Besides lowering the pH, lab act as pathogen competitors, antimicrobial compound producers and contribute to the depletion of carbohydrates substrates in favor of lactic acid production. 1, 39, 40, 41. A previous study performed by Coughlin et al. In raw goat milk underlines the ability of Enterococcus faecium to selectively inhibit Listeria monocytogenes and Clostridium butyricum by means of bacteriocins production, whereas none of the other indigenous labs suffered the effect of these natural antimicrobial compounds. 42. Lactococcus lactis and Enterococcus faecalis inhibited Staphylococcus aureus growth after 6H of incubation, most likely by means of acidification. Lactococcus garvii inhibited staphylococcal growth without acidification, suggesting acidification as one of the several defense mechanisms of the indigenous microflora against pathogen growth. 43. Other studies performed on the anti-listerial activity of the raw goat's milk microbiota indicate Lactococcus and Enterococcus strain as active players in Listeria monocytogenes inhibition by means of a novel nisin variant production. 44. Accordingly, a further study performed by Skiru et al. On raw goat milk microbiota selected 4 different Enterococcus strains for their ability to inhibit the growth of 21 Listeria monocytogenes strains and 6 Salmonella sp. isolates following bacteriocins production. 45. A research aimed at monitoring the microbiological quality of Caprino d'Aspromonte, a raw goat's milk cheese produced in Italy, demonstrated that strains of Lactobacillus paracasi subsp. Paracasi exerts a strong antagonistic activity against Escherichia coli through bacteriocin production, suggesting the indigenous microbial strains as a useful tool to control pathogen growth. 46. Additionally, in the same cheese, a natural reduction of coliforms and other hygienic indicator bacteria by means of the indigenous microflora was described during the ripening. Anyhow, improvement of the hygienic condition at milking, milk storage and all other processing steps was also suggested to reduce the pathogen contamination risk and improve food safety. 47. The adoption of high hygienic standards can also prevent the milk from pre-treatment processes, for example pasteurization, in favor to a rich and balanced indigenous microflora, thus preserve the typical flavor, texture and aroma of traditional cheeses. 20. 4. Cheese microbiome investigation in the omics era. Interest in understanding the origin, composition and role of the milk microbiota is steadily increasing in the last years, along with the consciousness of the role played by the microbiota in the development and diversification of the myriad of cheese varieties. Previous works already provided an excellent review of the latest omics technologies adopted for the investigation of the milk-associated microbial community. 48,49. In this section, we focus our attention on the investigation of the cheese microbiota and the major challenges the researchers have to face with, due to the changing consistency, texture and composition of the different cheese varieties. Methods adopted for the investigation of the cheese-associated microbiota changed from the classical cultivation-based approach to the most recent omics sciences. 50. In the past investigations, several achievements have been reached by means of traditional methods. For example, culture-based and molecular approach, especially in understanding the microbial species variation among cheeses varieties and between the diverse cheese-making processes. 51. Following the advent of the high-throughput sequencing platforms, investigation of the dairy microbiota, as well as the microbiota harbored in other ecological niches, has moved toward a renewed vision, employing a holistic conceptualization of the biomolecules responsible for the structure, function and dynamics of a given consortia of microorganisms co-inhabiting the same ecological niche. 52. Disciplines involved in the identification and quantitation of the biomolecules featuring the microbiota are therefore metagenomics, metatranscriptomics, metaproteomics and metabolomics, targeting respectively DNA, RNA, proteins, and metabolites produced by the whole microbial community. 53. The following sections highlight general guidelines, issues and disadvantages featuring the study of the cheese microbiota. Figures 1 and 2. 4.1. Cheese sampling. The first key step in microbiota investigation is represented by sampling strategy. Figure 1. Indeed, microbial biodiversity of the cheese microbiome is not evenly spread all over the cheese wheel. The cheese rind is an aerobic environment and is constantly exposed to the risk of contamination by external sources. The cheese core becomes an anaerobic environment during ripening, and it is certainly less exposed to further external contaminations sources. 54. Therefore, choice of the sampling area is strongly dependent by the aim of the study. Core microbiota is most commonly investigated in relation to its influence on the organoleptic composition of cheese, flavor development, and as a microbial fingerprint for differentiating closely related cheese varieties, whereas surface-associated microbiota is mostly investigated in relation to the effect of the manufacturing technology, ripening environment and its concern in cheese biosafety. 2,55. Sampling procedure varies greatly depending on cheese type, for example hard, semi-hard, soft. Besides the researcher's interest in investigating the whole microbiota or a subpopulation of the microbial community, rind samples are often collected by means of scraping using a sterilized razor blade. 56. Figure 1. Cheddar cheese rind has been sampled by collecting the samples at a depth of approximately 5 57. Another study collected the rind samples by cutting the first 2 thickness of the surface. 10 grams of the collected surface was subsequently homogenized by using a stomach-bag mixer registered. 58. Similarly, rind of Cota cheese was aseptically removed in a laminar flow cabinet and ground to 2 particles by means of a food processor. Particles were further subjected to stomach homogenizer, and 15 G of the homogenized sample was used to microbiota investigation. 59. Collection of core cheese samples expects the discard of the surface layer, approximately the first centimeter, in order to directly access the cheese mass and avoid cross-contamination from the surface-associated microbial community. Figure 1. Additionally, rind removal helps to uniform salt in moisture level differences existing between the exterior and interior regions. 60. Following this approach, Sunyos et al. Collected 10 grams of cheese core and then homogenized it in a stomach mixer. 58. Similarly, Escobar processed 15 G of core cheese obtained from a 3 kg piece previously stored at 70 degrees Celsius and thawed for 18 H at 4 degrees Celsius. 59. Herb cheese core has also been sampled in a similar manner. Cheese has been cut diagonally 2 times, in order to make accessible the core portion. 25 grams of the cheese heart was then collected and homogenized prior to proceeding to the total microbiome analysis. 61. Discrimination between surface and core microbiota became marked with the ripening process. In this view, sampling of the cheese precursors during the cheese-making process, for example curd, does not account for this discrimination, and sampling is much easily performed by means of a sterile punch, commonly inserted perpendicularly to center of the cheese and rotated 360 degrees to obtain a coring representative of the whole cheese mass. 2,6. Regardless of sample type, homogenization results in an even mixture of the sample components. Figure 1. Nevertheless, in the case of some very soft cheeses or fermented milk products, homogenization is omitted because of the loose texture of the dairy product, and samples are directly processed after collection. In a recent study performed on mozzarella, cheese has been directly frozen, (20 degrees Celsius, at the end of its production process, and only 50 (mg) of mozzarella have been aseptically cut out and directly used in the downstream analysis, 62. Also, in a study performed by Walsh et al., 20 milliliter of kefir was collected immediately after production and stored at 20 degrees Celsius until its direct processing, 63. Another important aspect to consider while collecting the samples for the cheese microbiota investigations rely on the preventive account for the natural variability occurring between microbial communities harboring, for instance, the same cheese variety and/or colonizing a cheese produced in the same dairy industry. To ensure a comprehensive investigation and accurate results, biological replicates are commonly employed while investigating cheese microbiota. The exact number of biological replicates is strictly dependent by the adopted experimental design, postulated hypothesis and future statistical surveys, but current literature report that at least 3 biological replicates are required to confidently describe the features of the investigated microbial community. Although, the involvement of a statistician is warmly suggested, 56, 57, 59, 62, 63. 4.2. Sample preparation. Once collected, samples are assumed to be representative of the entire microbial community harbored in the dairy product of our interest. However, along with the microbial fraction, cheese sample contains a wide array of molecules that interfere in the downstream processes of microbiota characterization. In this view, optimizing a sample preparation protocol is a fundamental prerequisite before applying any of the above-mentioned omics sciences. The main goal of the sample preparation protocol is to enrich the samples in the microbial fraction and virtually avoid the co-extraction of cells and biomolecules from the cheese matrix. Figure 1. Therefore, major challenge relies upon the fact that the reduction of the matrix interference is unavoidably coupled with the loss of target cells and/or biomolecules, in an extent that is strongly dependent by the type of matrix and the available technical equipment. 64. Isolation of the microbial fraction starts with the sample homogenization in the presence of a sterilized buffer that facilitates detaching of microbial cells from the matrix and prevent them from clumping together. Figure 1. Most commonly used buffer includes 2% Wv sodium citrate, 60, 65, 66. But Na2Ho4 and Tween20 might also be effectively used, 67. Contrariwise, other authors opt to homogenize cheese samples in sterile water, 3, most likely to avoid that buffer itself could interfere with the downstream analysis. Homogenization is followed by incubation under reciprocal shaking incubation, along with multiple steps of centrifugation, resuspension, interspersed with sonication bath incubations, in order to separate fats and other coarse material and physically disjoint microbial cells from the cheese matrix they are harbored. The microbial fraction is recovered in the aqueous phase and is finally pelleted by means of ultracentrifugation, around 13,000 times G, yielding the bacterial fraction of the sample in a dry and clean form, 68, 69. Figure 1. Once performed the common steps of sampling and sample preparation, diverse techniques and protocols are adopted for each of the above omics science. Following sections provide an overview of the single omics sciences adopted in the context of cheese microbiota investigation. Figure 2. 4.3. Metagenomics. Metagenomics investigation of the cheese microbiota aims to study the whole genome of the entire microbial community harbored in a given ecological niche, that is the cheese, providing information on the taxonomical biodiversity and potential metabolic feature of the microbiome. 70. The key step of the metagenomic investigation consists of DNA extraction from the previously enriched microbial fraction. DNA is subsequently subjected to sequencing prior to the step of bioinformatic data analysis. Figure 2. 4.3.1. DNA extraction. Extraction of the DNA is crucial since it must represent the heterogeneity of microorganisms composing the cheese microbiome, and must confidently mirror their relative abundance ratios. Among the diverse microbiota composition in the diverse cheese types, no unique protocol has been validated yet. Latest literature highlight that some research groups perform the DNA extraction protocol by using commercial kits. Of these, Power Soil DNA Extraction Kit, Mobio, 56,57, Power Food Microbial DNA Isolation Kit, Mobio Laboratories, 71, and Neasy Blood and Tissue DNA Extraction Kit, Kyogen, 58, are examples of the most commonly employed ones. A recent study on the Swiss Maasdam cheese performed the DNA extraction by means of the Wizard-registered genomic DNA purification kit, Promega, with some modification empirically set up by the authors for what concerns the cell lysis. 72. Use of commercial kits has the benefits of a fast and standardized procedure, enabling higher comparability between samples. On the other hand, the use of commercial kits is highly expensive and, to date, no kit specific for the cheese associate microbial community has been optimized. Other authors, instead, prefer the manual extraction since it allows to customize the protocol to better suits the own sample requirements. For instance, prior to inducing cell lysis, Escobar's Epidary suspended the microbial fraction in Nazi buffer, 100 trice pH 7.5, 25 mg Cl2 and 5 mca Cl2. Along with 1 U per milliliter of Nazi, in order to remove free DNA, mainly of bovine origin. Nazi must be inactivated, for example heat inactivation, prior to the microbial lysis, to preserve microbial-derived DNA. 59. Other studies resuspend the microbial pellet, prior to its lysis, in a 2.5% VV Triton X100 buffer, in order to dissolve casein Michael's residual from the cheese matrix. 57,73. Common cell lysis methods adopted in cheese metagenomics expect the use of enzymatic cocktails such as 10 mg per milliliter lysozyme in 100 trice HCl buffer and 50 and 10 U per milliliter of mutanol isonint buffer, 59,72. Nevertheless, other methods lysing microbial cells using chemical, phenol, chloroform, and isoamyl alcohol 25:24 to 1, respectively, detergents etc., and/or physical, sonication, heat shock, bead beating, methods, 72,74, are employed. Lysed cells are subsequently treated to remove endogenous interfering biomolecules such as RNA and enzymes, proteins that are readily removed by the respective addition of RNase and proteinase, for example proteinase K. Extracted DNA is finally precipitated by means of a high salt concentration, typically sodium acetate in 95% ethanol, but high concentrate NaCl solution, 6M, is also commonly used, 70,75. To maximize DNA recovery, the aqueous phase can be separately collected and another phenol chloroform isoamyl alcohol, 25:24 to 1 VV, respectively. Extraction may be performed, 75. Regardless of sample type and/or adopted sample preparation protocol, extracted DNA must meet requirements of quantity and purity set by the sequencing platform manufacturer. Eventually, a variety of commercial kit is available to yield purified DNA, even though it is often associated with a reduced final DNA concentration, 70. 4.3.2. Library preparation and metagenomics sequencing. Metagenomics sequencing is based on the so-called next-generation sequencing, NGS, platforms, among which 454 Roche pyrosequencing, solid, and Illumina sequencing by synthesis are the most frequently adopted ones, 53. Protocols for library preparation and sequencing reagents are strictly dependent on the sequencing technology and the adopted sequencing equipment, 64. Studies based on amplicon sequencing require a previous PCR-based amplification of the genomic fragment of interest. Commonly, hypervariable regions of the 16S gene are selected and amplified to account for the bacterial diversity, whereas 18S gene and its regions are generally targeted to refer to yeast and fungal composition respectively, 70. Amplicon libraries and/or the total extracted DNA, whole genome shotgun analysis, are subjected to an indexing PCR, aimed at tagging DNA fragments with adapters for the subsequent sequencing step. Figure 2. Here, primer sequences, amplification condition, and template purity and concentration vary independence of the sequencing platform and technology. In the fungal microbiota investigation performed by Sunez et al. On artisanal cheese, 5.8 CITES2 RDNA PCR libraries were prepared using universal primers with Illumina overhand adapters targeting the ITS2 region. Amplicons were purified by using the Egencourt AM Pure XP beads kit, Beckman Coulter, prior being subjected to a second indexing PCR according to Next-RUXT index protocol, as expected by the Illumina MeSeq v3 platform. 58. Similarly, the recent study of Bututsi et al. Performing the whole metagenome shotgun sequencing of the rind-associated microbiome expect the library preparation according to Next-RUXT DNA guide released from the manufacturers, while sequencing was performed on the Illumina MeSeq and Illumina NextSeq sequencing platforms. 57. Unlike metagenomic analysis performed by Dugat-Boni, instead, has been run in the solid, applied biosystem, sequencing platform. Here, the DNA libraries were constructed from 3 μg of total DNA by using the solid fragment library construction kit, applied biosystems, and then barcoded with the solid fragments library barcoding kit, prior being sequenced by means of the solid 4 system. 73. 4.3.3. Metagenomic studies on cheese microbiota. One of the first cheese metagenomics studies was performed by Wolf et al. 2014, investigating the cheese rinds in 3 diverse contexts, that is bloomy rinds, natural rinds and washed rinds. The study provides a list of the putative functions attributed to the sequenced genes and hypothesizes the potential contribution of some psychotrophic bacteria in the development of the typical flavor of 137 different cheese types from 10 countries. 56. In a study by Marino et al., metagenomics was used to discriminate between microbiota of cow and buffalo mozzarella. Authors suggested the suitability of the metagenomics as a tool to control milk origin since 2 diverse microbiota composition were clearly observed. Cow mozzarella is featured by a higher bacterial diversity, 2-tailed t-test for JAW1 diversity indexes, P10-4, goods coverage, P equals 0.0255, and a higher load of psychotrophic bacteria such as Anoxibacillus spp, Brocothrix spp, Flavobacterium spp, Pseudomonas spp, Schwanella spp, and Thermos spp. Buffalo mozzarella is, instead, characterized by an increased abundance of Lactobacillium streptococci. 62. The fungal microbiota of Tom Dorquise, an artisanal cheese produced in the north of France, was assessed on the basis of metagenomics. Here, the fungal microbiota is dominated by Yarrow politica, however, the presence of other unusual yeast strains not originating from the starter culture is also underlined. 58. In another study, Escobar-Zepido et al. Applied metagenomics to investigate the metabolic potential of the microbial community harbored in the Mexican Cota, highlighting a potential concern of the cheese microbiota in the metabolism of branched-chain amino acids, free fatty acids and bacteriocins production. 59. Acknowledged the bulk of additional information provided by the application of the metagenomics, the study of the effective functions performed by the microbial community as well as the complex network of interactions occurring between microorganisms cannot be elucidated only by the putative functions and metabolic potential provided by the DNA-based methods. Moreover, metagenomics is unable to distinguish between live and dead cells, that is metabolically active and inactive, respectively. Owing to these reasons, a valid strategy to investigate the effective functional profile of the cheese microbiota is to evaluate the gene products, RNA transcripts, transcriptomics and metatranscriptomics, and proteins, proteomics and metaproteomics. 50. 4.4. Metatranscriptomics. Metatranscriptomics is an emerging discipline based on the NGS platforms, aimed at investigating the gene expression of the whole microbial community. Although metagenomics provides information about all the potential functions a given microbial community is capable of, it does not provide indications on the effective gene expression, or gene dynamics, of the microbial community under determined circumstances. 76. Nowadays, the gold standard technique in studying the transcripts of a whole microbial community is represented by RNA sequencing. Although the sequencing technology is the same adopted in metagenomics studies, key differences are represented by the selective removal of the interfering nucleic acids. For example, DNA, tRNA, RNA, and the reverse transcription of mRNA to cDNA prior to library production and its subsequent sequencing. 52,77. Figure 2. 4.4.1. RNA extraction. As for metagenomics, the microbial fraction is subjected to RNA extraction by using either commercial kits or lab-assembled protocols. In a recent study, massive parallel 16S gene tag sequencing has been employed to study the bacterial diversity of brines used for cheese making. Microbial fraction extracted out of 50 ml of brine has been subjected to RNA extraction by using the RNeasy mini kit, QIAGEN 62. Similarly, another study investigated an Italian hard cheese using 16S-based approach, performing the RNA extraction via MasterPure Complete DNA and RNA purification kit, Epicenter. This kit was coupled with an additional step of DNA removal by means of turbodenase, ambient addition. Also, a control for the effective DNA removal is necessary, for example by PCR, 6. In another study on cheese metatranscriptomics, Deru et al. Provided a comprehensive lab-assembled method for high-quality RNA preparation. Here, the previously extracted microbial fraction is resuspended in RNA Protect bacteria reagent, QIAGEN, to prevent fast RNA degradation. The cell suspension is treated with a lysis buffer containing 10 mg per milliliter of lysozyme, 3 mg per milliliter proteinase K, and 100 U per milliliter of mutanolysin. Microbial lysis was also ensured by the bead beating treatment, in the presence of triaryl reagent, invitrogen. The lysate is extracted with chloroform and RNA is subsequently precipitated by the addition of 80% ethanol prior to being subjected to Neasy mini kit, QIAGEN, for total RNA purification. Ribosomal RNAs are finally removed out of the total extracted RNA by means of Ribosero commercial kit, Epicenter, by following the manufacturer's instructions. 72. Adopted protocol enabled a thorough investigation of the mast and cheese metatranscriptome, elucidating the metabolic contribution of the microbiota members to cheese flavor throughout the whole ripening process. Interestingly, Dugat-Boni, 73, proposes an alternative RNA preparation protocol directly in the cheese samples, without the prior separation of the microbial cells. Here, 500 mg of finely ground cheese samples are directly subjected to bead beating in the presence of triaryl reagent. Homogenized samples are then subjected to mild centrifugation, 4000 times G, in order to separate the organic phase, containing the fat, from the aqueous phase, containing the RNAs among the other molecules. The aqueous phase was extracted by means of two further passages, the first with chloroform and the second with phenol chloroform isoamyl alcohol, and extracted RNA was then purified with Neasy spin columns, QIAGEN, 78, 4.4.2. Retrotranscription, library production and sequencing. Extracted RNA is subsequently controlled for its quality and quantity before being retrotranscribed to cDNA. Most commonly used approach to evaluate RNA concentration and quality rely on the nanodrop UV-Vis spectrophotometer and via quantitative PCR performed using 16S RNA primers, 62, 73, 78, 79. But other methods also exist. For example, quid fluorimeter, invitrogen. RNA retrotranscription is rather commonly performed by using specific kits compatible with the subsequent sequencing platform and the desired sequencing chemistry. Figure 2. In the study of Deru et al., RNA fractions were amplified as cDNA and libraries were constructed by using Ovation RNA-seq system v2, new gen technologies. Obtained libraries were sequenced by using the solid 5500 XL, Life Technologies sequencer with single-end sequencing method, 75 BP single-read chemistry, using non-strand specific protocol, 72. Differently, Dugat-Boni used 200 to 500 ng of total RNA to produce cDNA libraries using a solid whole transcriptome analysis kit and were barcoded with the solid transcriptome multiplexing kit, 73. Instead, the study of Alessandria on hard cheese investigated by 16S-based approach produces the cDNA library by using Maloney murine leukemia virus, MMLV, reverse transcriptase, PromEGA. Copy DNA templates are subsequently amplified in the V3 hypervariable region of the 16S gene. However, no sequencing of the resulting amplicons was performed. 6. 4.4.3 Metatranscriptomic studies on cheese microbiota. First, cheese-related transcriptomic investigations focused on the metabolic featuring of a selected number of microorganisms, mainly bacteria, presumed to play an important role in the dairy industry under several points of view, including fermentative capability, biosafety, flavor development, 53, 96, 97, 98, 99, 100. These studies provided a comprehensive elucidation of the metabolic response of singular microorganisms under diverse experimental conditions. Nevertheless, very few studies are, to date, performed as metatranscriptomic investigation, that is regarding the whole microbial community harbored on cheese. This is most likely due to the methodological and technical issues the researchers have to face, including the development of a common protocol that evenly extracts all RNA transcripts from all species and also prevents them from degradation. 86. In a recent study, Philippis et al. Used metatranscriptomics to investigate the effect of temperature and humidity on the Casiocavallo Solano microbiota. Gene expression profile revealed that higher ripening temperature is associated with an increased expression of genes involved in proteolysis, lipolysis, fatty acid metabolism and amino acid metabolism. This overall gene expression profile resembles the expression pattern of the same cheese samples ripened at a lower temperature but for a longer ripening time. On this basis, authors suggested the increase of the ripening temperature in order to benefit operating costs and improve turnover in ripening room without affecting the cheese flavor and aroma. 87. In another study, metatranscriptomics was applied to investigate surface ripening by means of a mixed starter culture composed of Staphylococcus thermophilus, Lactobacillus delbrueckii spp, Bulgaricus, Geotrichum candidum, Dibromyces hansenii, and Brevibacterium aurantiacum. Evidence of this study describe an upregulation of Staphylococcus thermophilus genes involved in signal transduction, protein folding, sorting, degradation. Expression profile of Lactobacillus delbrueckii highlights its major concern in nucleotide and lipid metabolism, but also transcriptional and translational activities were highly represented. Geotrichum candidum shows an expression profile associated with carbohydrate, lipid, and amino acid metabolism, whereas Dibromyces hansenii is involved in the metabolism of other amino acids. Both yeasts registered a decreased expression of gene associated with nucleotide metabolism. 88. In a previous study, Lessard et al. Employed metatranscriptomic on camembert cheese to profile pathways associated with the production of the typical cheese texture, flavor and aroma. This study found that Geotrichum candidum and Penicillium camembert have a pivotal role in ripening and the development of the sensory characteristics of this cheese. 85. 4.5. Metaproteomics. Targets of metabolomics are the RNA transcripts, typically mRNA. However, it has been evaluated that some discrepancies occur among mRNA and protein abundance due to the variable degradation time of the transcripts, allosteric changes in mRNA, variable rate of protein translation and possible post-translational modification. 50. Thus, analysis of expressed protein repertoire is of fundamental importance to fairly assess effective functions of the cheese microbiota. 48. Figure 2. Only a handful of large-scale metaproteomic studies have been published so far and, to the best of our knowledge, only a single metaproteomic study was performed so far in the field of cheese microbiota. 3. This is most likely due to the challenges metaproteomics researchers have to cope with, regarding, for instance, the heterogeneity of organic and inorganic contaminants arising from the cheese matrix, high biodiversity of microorganisms colonizing the cheese and the over and under representation of microorganisms and proteins that make difficult a comprehensive representation of the cheese-associated microbiota metaproteome. 89. Once extracted the representative microbial fraction, common step to all described omics disciplines, a standard metaproteomics experiment consists of 3 basic steps. I. Protein extraction and purification. 2. Protein denaturation and reduction. 3. Protein prefractionation, enzymatic digestion, and MS analysis. 90. 4.5.1. Protein extraction and purification. To achieve protein extraction, the first step is to lyse the microbial cells by means of detergent-containing buffers. For example, STS, sonication, French press treatment, or a combination of these methods. In the pioneering study performed by Soggy et al., the microbial fraction isolation and protein extraction were performed at once. Here, a small amount, 400 mg, of finely grated cheese, was resuspended in milky water, sonicated and stirred before being centrifuged. This enables the further depletion of caseins, that is the most abundant class of proteins of milk and cheese, that would otherwise interfere with the downstream analysis, besides masking the identification of the other less abundant proteins of microbial origin. Other methods to deplete the casein fraction include the use of hexapeptides library and are reviewed by Roncourt et al. 48. Before proceeding with the further analysis of the extracted proteins, interfering compounds of microbial and or matrix origin such as nucleic acids, lipids and polysaccharides, have to be removed. To this purpose, nucleases, for example benzonase, are commonly added to the cell lysate, along with protease inhibitors, for example PMSF, to prevent protein degradation by means of endogenous proteases. 48,90. Moreover, the addition of cold trichloroacetic acid or acetone results in the precipitation of the extracted proteins, thus its physical separation from all other interfering molecules. Protein purification may also be achieved via 1D-STS page. This is the common choice of the so-called gel-based proteomics analysis, where the gel electrophoresis is also used to perform the protein prefractionation. The gel-free proteomics, instead, perform the protein peptides prefractionation by using single or multidimensional LC. Major steps in gel-free and gel-based approaches are provided in Figure 3. Alternatively, commercial kits are also available to ensure the purification of the extracted proteins, for example 2D cleanup kit, Biorad. Nevertheless, unlike DNA extraction, no effective kit for the whole metaproteome extraction has been so far developed. Therefore, care must be taken while optimizing the protein extraction protocol, since it should be as efficient, non-biased, and reproducible as possible. In addition, it is crucial to avoid the addition of organic or inorganic compounds, solvents that might interfere with sequential protocols and mass spectrometry, MS, measurement. 90. Figure 2. 4.5.2. Protein denaturation and reduction. Prior to their MS-based measurement, purified proteins are denaturated to break intra- and intermolecular interaction. Linearized proteins are then reduced and alkylated to prevent the reversibility of the molecular rearrangement. This can be achieved by employing buffers containing catropic, for example urea and or thera, non-ionic and or zwitterionic detergents, for example Triton X100, or CHAPS, ionic detergents, for example STS, reducing agents, DTT, dithioerythritol, or tributylphosphine. Alkylation agent most commonly used is iodoostamide, IAM. At this stage, either gel-based or gel-free approach can be distinguished depending, respectively, on the direct in solution or in gel occurrence of denaturation, reduction, alkylation and the following digestion of the proteins. Although scarcely automatable and of difficult handling, the gel-based approach relies on an impervious environment, protecting the sample from external contaminants. 90. Figure 3. In the gel-free metaproteomic investigation of Grana Padano cheese, approximately 50 micrograms of the purified proteins were resuspended in a denaturing buffer composed of urea, 8M, in 100 ammonium bicarbonate. Proteins were subsequently reduced and alkylated by sequential incubation with DTT, 10 DTT in 50 ammonium bicarbonate, and IAM, 55 IAM in 50 ammonium bicarbonate. 3. 4.5.3. Protein prefractionation, enzymatic digestion, and MS analysis. Prefraction of proteins prior to MS analysis results in reduced complexity of the sample, thus a higher, and more accurate, protein identification rate. Protein prefractionation may occur via gel electrophoresis or LC. Figure 3. In the latter case, the combination of various separation criteria such as strong cation exchange, SCX/reversed phase, RP, resulted in a very significant increase of the resolving power, suitable to address high complexity samples. 90. In our reference study, a bidimensional chromatography, SCX/C18, approach has been used to resolve the sample complexity. 3. In the gel-based approach, sample prefractionation is performed via electrophoresis. For several years, 2D STS has been considered the gold standard of proteomic investigation. However, the recent technological progress in the field of LC-MS enabled the development of a 1D STS followed by in-gel digestion of trapped proteins and peptides resolution by RP chromatography. This hybrid approach combines the cleaning properties of the gel electrophoresis along with the high resolution of LC. 90. Figure 3. Regardless of the adopted proteomic approach, peptides generation occur by means of enzymatic digestion. Trypsin is the most commonly adopted enzyme, but chymotrypsin is also widely used in proteomic investigations. 90. Once reduced sample complexity, peptides, bottom-up proteomics, are finally subjected to the MS measurements. Newest automated approaches have the LC system directly connected to the MS apparatus, thus eluting peptides are automatically injected to the ionization source of the mass spectrometers. Most commonly used ionization techniques in proteomics experiments are MALDI and DESI. Ion sources can then be coupled to various mass analyzers, most commonly DOF or ion traps. Nevertheless, metaproteomic investigation of complex samples such as the cheese microbiota requires higher performing machines such as tandem mass spectrometers MS study performed by Soghiu et al. Measured the tryptic peptides on Amazon Speed ETD ion trap MS/MS, Bruker, but also other state-of-the-art mass spectrometers such as hybrid quadrupole DOF analyzers, FTICR mass spectrometers and LTQ-Orbit-RUP mass spectrometers allow rapid and sensitive targeted MS/MS on LC timescales and highly accurate mass determination in the low ppm to sub-ppm range. 3,48,90. 4.5.4 Cheese microbiota studies involving proteomics, metaproteomics. Recently, a proteomic approach has been used by Zanni et al. To quantitatively investigate the protein repertoire as expressed by Lactobacillus delbrueckii subsp. Bulgaricus and Lactobacillus delbrueckii subsp. Lactis, both isolated from a traditional fermented cheese. This study highlighted that Lactobacillus delbrueckii subsp. Bulgaricus was the sole involved in amino acid metabolism, whilst Lactobacillus delbrueckii subsp. Lactis expressed a higher abundance of proteins related to maltose and glutathione metabolism. 91. Other similar studies have been performed to feature lactic acid bacteria and the mechanisms exerted to survive under a plurality of changing environments. 57,74,75,88. Although these studies are of scientifically sound, they do not provide desirable knowledge for an exhaustive investigation of the cheese microbiota and its dynamics. For this purpose, metaproteomics studies are required. A recent metaproteomics study has been performed by Soghiu et al. To unravel the functional dynamics of the Grana Padano-associated microbiota. In this study, the cheese samples were experimentally supplemented with varying load of Clostridial spores and further supplemented, or not, with lysozyme. Identified protein repertoire enabled the elucidation of the metabolic impact the lysozyme addition has in regard to the Clostridia associated with late blowing defect. Also, the assessment of the active bacterial fraction on the basis of the identified proteins revealed that lysozyme addition has a major influence on the bacilli class. 3. 4.6. Metametabolomics. Cheese metametabolomics investigations would aim to depict the overall array of metabolites produced by the consortia of microorganisms associated with cheese, enabling their link back to the ongoing physiological processes. 92. Similarly to transcriptome and proteome, the metabolome is very dynamic, therefore, its investigation under diverse circumstances might provide precious information on the causality of a given phenotype or, inversely, the systemic effects of specific substances and/or experimental treatments on the associated microbiota. 52. Nevertheless, metabolomic investigation so far performed in the field of dairy industry aimed at investigating the dairy product composition and/or few microbial isolates. None of these studies focuses on investigating the functional dynamics of the microbial community harbored in the dairy products. 50. We strongly believe that constant progress of the omics sciences along with the increasing interest in characterizing the cheese microbiota will soon result in a widespread application of metametabolomics studies of the cheese microbiota. 4.7. Bioinformatics. Although the progress in the field of NGS benefited microbiota characterization under diverse points of view, it also results in an enormous amount of data, which treatment and integration require strong computational efforts. Metatranscriptomics and metaproteomics are strongly dependent on the quality and availability of the metagenomes used as a blueprint for the mRNA and protein identification, respectively. Figure 4. Although the huge advances in the sequencing technologies, it is still difficult to obtain complete strain-specific genomes and, in the context of the microbiota evaluation, only partial population genomes are generally available. Despite this limitation, a wide number of bioinformatics tools is nowadays dealing with metagenomics sequences, enabling an integrative approach between omics data. Commonly used metagenomics tools are MEGAN, Metagenome Analyzer, IMG/M, Integrated Microbial Genomes/Metagenomes, and MGRAST, Metagenome Rapid Annotation using subsystem technology. These, although based on different algorithms and slightly different statistics, enable the taxonomic and putative functional annotation of the metagenomic sequences, and allow the comparative evaluation of multiple datasets. In addition, MGRAST and IMG/M are also used as data repositories and, along with NCBI, National Center for Biotechnological Information, and EBI, European Bioinformatics Institute, represent the biggest data repositories currently available. 64,93. A typical LC-MS/MS run from a metaproteomics investigation generates hundreds of thousands of fragmentation spectra. Clearly, this huge amount of data cannot be manually elaborated, and a wide variety of bioinformatics software and tools is nowadays available to translate the myriad of spectra into a meaningful output that is both concise and informative. 94, Figure 4. To this purpose, commonly used search engines are OMSA, Open Mass Spectrometry Search Algorithm, X, Tandem, Mascot, Andromeda and Sequest. Major tasks of these tools include quality filtering of the raw MS spectra, peptide spectrum matching and scoring, protein database searching, data mining, and graphical representation of the obtained results. 95. Similarly to metagenomics tools, proteomics, metaproteomics committed tools are based on diverse algorithms and statistics, resulting in varying performance that, for each tool, depends on several factors such as sample complexity, separation efficiency, and quality of the reference database. 64, Figure 4. The ideal metaproteomic database comprises all potentially expressed proteins in the sample of interest and lack of additional spurious sequences. This is rather challenging for the metaproteomic investigation of complex samples such as the cheese microbiome, mostly because of the highly dynamic and heterogeneous microbial composition, other than the great variability occurring between and within cheese types. To date, the best, and more accurate, solution expect running the database-dependent search of the generated spectra against a metagenomic matched database. However, this results in a conspicuous increase of the analysis costs, making this strategy unsuitable for widespread metaproteomics investigations. 96. Customized databases are often used to simulate a metagenome database. 97,98. Although this results in improved peptides and protein identification rate, it might suffer from the reduced coverage of the in-house metagenome database compared to the real metagenome sequences. Also, arbitrary choice of the included taxa hurdle results comparison of different studies. 99. In this view, in a study aimed at investigating the human and mouse gut microbiota, that is traditional examples of complex samples, Zhang et al. Implemented the metaproteome identification and quantification approach, MetaPro-IQ. This approach expects a first unfiltered database search against the close to complete human or mouse gut microbial gene catalog. Here, exported database is assumed to be a list of all the potentially expressed proteins and is further hyphenated with reversed sequences, enabling the run of a second target decoy database search. Common database-dependent searches performed in metaproteomics studies. Resulting datasets for each sample are merged and deduplicated, enabling a third database-dependent search against the in-house non-redundant database. This approach has been proved to ensure comparable performance with matched metagenome strategy, 96, even though benefiting of a priori close to complete gene catalog availability. Proteomic, metaproteomic data repositories can be distinguished on the basis of the annotation type. UNI-PROT-KB, for instance, is further divided into Swiss PROT and TREMBL. The first comprises the only entries whose annotation has been reviewed and manually annotated, whereas TREMBL entries are not reviewed and only automatically annotated. Other data repositories commonly used are Kyoto Encyclopedia of Genes and Genomes, KEGG, Gene Ontology, Go, BioCarta. These provide information concerning the subcellular localization, biological process and molecular function for each of the listed proteins. Moreover, KEGG integrates all this information and organize them as biochemical pathways that fall into defined biological groups. Recently, IMTLab, a web-based platform, has been released for the metaproteomics data analysis, 100. Diverse other software and web-based applications are nowadays available for the functional annotation of the omics data such as WebMGA, Cytoscape, IPath, David, Metaproteome Analyzer and others, as reviewed in 64, that retrieve and integrate functional annotation from diverse sequence repositories to provide a more comprehensive functional depiction of the queried dataset. In light of the above, it is easily understandable that metatranscriptome and metaproteome data are strongly dependent on the availability and quality of the metagenomics sequence, Figure 4, but research is to prevent the total dependency of metaproteomics from the metagenomics sequence availability are currently ongoing. 52,94,101, 5. Microbiota-dependent nutraceutical properties of dairy products. Diverse aspects of the milk microbiota are nowadays the aim of studies, including investigating the role of the milk microbiota in biosafety, flavor development and technological aspects. In a recent study, Walsh et al. Adopted a novel approach to investigate kefir, a health-promoting beverage obtained by fermentation of milk by means of a consortium of microorganisms. Here, a metagenomic approach has been applied to investigate the microbiota composition and gene content, whereas GCMS was applied to characterize volatile profile. Altogether, both methods complement each other with the aim to provide further knowledge on fermentative process, flavor and biosafety of kefir and other fermented dairy products. 63. In a similar study, a whole genome shotgun sequencing and GCMS approaches were complementarily adopted to evaluate the microbial population and gene potential of surface-ripened cheese obtained by using two diverse smear culture mixes. The study confirmed the diverse microbiota in ripening stage depending on the addition of the two diverse mixes. Also, GCMS analysis enabled the link of the identified microbial species to their specific metabolic product, elucidating the role of the microbiota components in the flavor development in the finished product. 57. Large-scale dairy industries make use of strictly controlled starter culture blends in order to ensure the production of a standardized product and optimal processing attitude. In this view, Lactobacillus casei, Lactobacillus paracasi, Lactobacillus rhamnosus, and Lactobacillus plantarum are often metabolically characterized in order to elucidate their contribution to flavor development in milk-derived products. 53,54,102. Other studies extend their interest in screening the whole lactic acid-producing bacteria fraction in order to identify the optimal mixture of microbial starter, in terms of both fermentative and probiotics activity. 55,64. Karami et al. Investigated the antagonistic activity of lactobacilli strains isolated from various dairy products and, owing their health-promoting effects, suggested a potential implication of Lactobacillus alimentarius, Lactobacillus sake and Lactobacillus collonoides in the production of various pharmaceutical products and milk-derived foods, including functional foods. 103. Besides lactobacilli, Other bacterial strains commonly found in the microbiota of dairy products have shown interesting technological and probiotics activities. For instance, genomic investigation of Propionibacterium fudenreicii and Propionibacterium acidipropionaceae revealed their potential to adapt and survive in diverse environment and conditions, most likely due to propionic fermentation capability and the ability to use various carbon and energy sources, besides the recent evidences of probiotic features, as supported by their ability to benefit gut microbiota and host metabolism, physiology and immunity. 104,105. Also, composition and dynamics of milk are typically investigated in the attempt to designate autochthonous microbial community, elucidate its features, and link them with specific aspects such as a designated geographical area or productive practice, specific cheese composition and/or the unique gustatory and olfactory properties of specific derived products. 106. 107, 108. In addition to the food industry and all its surrounding aspects, milk microbiota is thoroughly investigated in relation to its nutritional value and the multifaceted health-related benefits in both human and animals, primarily arising by its direct impact on the gut microbiota. Recently, Bautista Gallego and colleague evaluated the effect of consumption of fermented milk in healthy volunteers, underlying the impact of this dairy product on the gut microbiota composition. Also, administration of a Lactobacillus rhamnosus strain isolated from ripened cheese resulted in an increased level of the Prevotella fraction along with a reduced portion of the bacteroides detected in the human fecal microbiota. 109. On the basis of the fact that probiotics can indirectly benefit the central nervous system via the gut-brain axis, May, et al. Examined the role of Lactobacillus helveticus, a common milk probiotic, in the subchironic and mild social defeat stress model mice, highlighting improvement of anxiety or depressive behaviors of mice, attributable to the administration of the milk probiotic. 110. An interesting review of Ritzeli and Biver summarizes the recent studies on dairy products and their beneficial effects on bones as provided by means of their composition, for example vitamin, proteins, minerals etc., prebiotic source, for example inulin, and non-ultimately the associated microbiota. 111. Beneficial health effects of milk microbiota are also exploited in the field of animal health and wellness. A recent study of Kim et al. Experimented, for the first time, the potential application of kefir as a probiotic supplement for dogs, highlighting a successful modification of the canine gut microbiota without registering any evident side effect. On this basis, the authors suggested the use of kefir as a novel approach to provide probiotic to improve quality of life of dogs. 112. 6. Conclusion and future perspectives. Milk is a heterogeneous body fluid occurring in nature to address the nutritional and defensive needs of the mammal's newborns. Owing to its biological potential, milk is a precious product for the dairy industry where it is mainly transformed into cheese and a diverse array of other milk byproducts. Microbiota of milk and its derivative, especially raw milk and its byproduct, is rather heterogeneous and multifaceted. Recent groundbreaking evolution of the omics sciences along with bioinformatics data analysis opened new avenues in the investigation of milk microbiota, shedding light on a finely tuned series of microorganisms, biochemical pathways and a high complexity of dynamic interconnections than ever thought. Since then, several studies have been performed to elucidate various aspects of the intricate interconnections existing between bacteria and other microbiota members. Employing omics sciences for evaluating the effects of bacteriophages in the starter culture adopted in the large-scale dairy industry is to date a hot topic, representing the most relevant biological issue in the massive dairy industry. Besides, understanding origin, role, risk and benefit for each of the microbial species harbored in such a complex ecological niche is also widely explored. Nevertheless, many pieces of knowledge and information on the milk-associated microbial community are still missing. At this purpose, it is strongly believed that the holistic conceptualization of the milk microbiota is the key to retrieve high and applicable knowledge under the diverse field of application and, considering the economic impact associated to the optimization of the milk microbiota, we are confident that microbiota investigation of dairy products will markedly increase in the near future.
And now it's time for you to sleep, if you're not asleep already. Rest well, curious person.